Welcome to STAAR Mix 4, Cell Processes and Homeostasis. This is best viewed after Mix 3. Look over the words that you see here carefully. If there are any that you do not recognize, write them down for a review later. If you don't recognize them, take a moment to try to remember what they mean and tell yourself again what you have learned about those words. A lot goes on in your cells. Your cells are responsible for creating proteins and replicating new cells. But they're also responsible for metabolizing the food energy and carrying out gases throughout your body, like oxygen and carbon dioxide. You're a large, intricate machine, but you don't work without the tiny little processes that happen in every one of your cells. Or for that matter, the tiny little processes that happen in plant cells that you're going to eventually eat. It's important that the cell is supplied with all of the substances it needs. For example, that the cell gets oxygen, and that the waste substances like carbon dioxide or substances for export leave the cell. There are various processes by which this can happen, and we're going to talk about each of them. The first thing that I want to show you is how molecules move naturally. If you've struggled with this tough stuff, it's important to start from the beginning. Diffusion is the idea that all molecules have constant movement, and that molecules will tend to move from an area of high concentration to an area of low concentration. Molecules do this regardless if they are in your body, or in regular water, or in the air around them. It's just what they do. Molecules tend to spread out. They're going to always move down the concentration gradient toward areas of lesser concentration. Think of something like food coloring that spreads out in a glass of water, or air freshener that's sprayed in a room. So what is this concentration gradient? Well, believe it or not, you've experienced this anytime you walk into a room with a strong odor. Concentration gradient is basically the difference between the concentrations in any given space. So when the smell is released in one area, it's very strong, and it seems like it comes at you. Well, there were no odor molecules where you were when the smell was released. In any solution, whether it's a solution of different gases like CO2 or O2, or in a solution of liquids like salt and water, there's always going to be a ratio of these ingredients. For example, in the GIF that you're seeing here, you have a ratio of the normal air molecules indicated in blue, and you have the ratio of whatever the smell was coming out of the bottle in green. The concentration gradient tells the green molecules that it's free to leave the bottle and explore the outside air, just like releasing a foul odor. This diffusion is the process that is used to move oxygen and carbon dioxide in and out of your cells. The molecules will move from where there's a high concentration to where there's a lower concentration, i.e. they diffuse down the concentration gradient. The blood system in humans is continuously bringing more oxygen to the cell and taking carbon dioxide away from the cell. This is going to maintain a high concentration gradient. Since the movement is always going down the concentration gradient, it requires no energy. The small molecules pass from one side of the membrane to the other by moving between the lipid molecules. All cells have a structure that plays an important role in the process of diffusion, but it also has many other properties that give it the ability to determine the balance between the outside of your cell and the inside of your cell. This ability is called selective permeability. While things like water and gases will move based on the concentration gradient, other things need a little bit more help. It's the job of the plasma membrane to maintain homeostasis. Your plasma membrane has three distinct jobs. First, it allows nutrients and water into the cell. Second, it's going to keep the levels of the nutrients and water at a balance. If there are too many, it's going to let some out. If there's not enough, it's going to let some in. Third, it's going to allow waste products to leave the cell. The basic makeup of the membrane is fairly simple. As you get further into biology, it'll get more complex, but we're only going to go over the basics here. The plasma membrane is also known as the phospholipid bilayer. Bi means two. There are two layers of the phospholipids. Remember, a phospholipid is a lipid that's made out of a phosphate molecule, here's the phosphate, and a glycerol molecule, and two fatty acid chains, and here's your two fatty acid chains. The head of the phosphate molecule is attracted to water, whereas the two fatty acid tails are repelled by water. There's watery environments inside and outside of the cell. Therefore, the phospholipid layers are going to look like a sandwich with the head ends facing the watery environment that they are attracted to, and the tail ends are going to be inside the sandwich, away from the water. So here they're repelling, or they're attracting the water here, bringing the water in. Here they're not liking the water, and so they're pushing it back out, where it's again attracted. This gives a good push and pull to the water, so it causes the water to constantly flow in and out of the cell. There are other parts of the plasma membrane, including the cholesterol molecules. These are going to be found throughout the plasma membrane, and they basically aid in lubricating the plasma membrane. You can kind of think of it like pasta and butter. If you were to make a pot of pasta and not put any butter in it and leave the pasta sitting there for a while, it would get really hard, and if you tried to pull the noodles off, they wouldn't come off. But if after you made the pasta and you drained out the water and you put some butter in it, later on, once the pasta cooled, you would be able to move the noodles around, and that's pretty much the way cholesterol works. Proteins are also going to span the entire plasma membrane. You can see a transport protein here, it's indicated by the arrow. They're going to help the membrane to be selectively permeable. They're going to move the substances that need to come into the cell, and they're going to move the waste materials out. Remember, not all things are small, and so the larger materials, or the materials that can't be absorbed by the lipids, they're going to have to go through protein channels. These channel proteins, they line a water-filled pore in the membrane, so water-soluble molecules can easily pass through. Different channels are going to allow different substances to pass through the channels, i.e. they're selective, but some channels are gated, so they're only going to open whenever they're appropriately stimulated. Any type of diffusion is going to be passive, but there's several different forms of diffusion. The one I want to look at here is the passive diffusion of gases, and this happens all the time in your body. So, for example, the lung surface area is made very large by these little tiny structures deep inside your lungs called alveoli. The difference in the concentration is maintained by the breathing. You inhale oxygen in the atmosphere. When you inhale, you bring in a lot of oxygen into your bloodstream, which is carried directly to your blood cells through these little tiny surfaces. And what you're seeing right here is one teeny tiny alveoli, the skin of it. This skin here is actually one cell thick. It's really, really tiny. So you can see this person is inhaling oxygen. As these gases come in contact with the cell, coming through here one cell at a time in this image, they start to turn red. The redness of the cell says that the gas has passively entered the cell. On the flip side of this, carbon dioxide will passively leave the cell, entering the alveoli. I'll do that in a different color. The purple you see here is carbon dioxide, and that's going to leave the red blood cell and go back into the lungs for you to exhale. So every time you inhale, you're taking in oxygen, and you exhale, you're removing carbon dioxide. You are actively participating in passive diffusion at the cellular level. Osmosis is the passive transport of water only. This is a special case of diffusion in which we're only concerned with the movement of water. So up until now, we've been looking at the passive diffusion of just about anything. It didn't matter the size or where it was at. But with osmosis, only water can move. So for example, if you have two different solutions and they're separated by a semi-permeable membrane, which in this case, we're working with your cell membrane, and only certain size molecules can go through, this means you're not going to be balancing everything else out just by the numbers. You're going to be balancing out based on the ratio of water to whatever solute was in it, like let's say it's salt or sugar. So let's take a look at this image that we're showing here. On the left-hand side here, we have just pure water. We have our selectively permeable membrane here. Water is designated by the blue circles, and in this case we're dealing with sugar. So we have a formula over here which has sugar, it's probably glucose, and water. But notice that there's only sugar on the right-hand side. So the water gets pulled into the right-hand side in order to balance out the ratio. The same kind of thing is going to happen in your cells. So the left-hand side, we would say it is low in sugar concentration, but high in water concentration. On the right-hand side, we would say it is high in sugar concentration, but low in water concentration. And that's going to pull the water over. Being able to predict which way the water will flow is called water potential. Water always moves from a region of higher water potential to one with lower water potential, or basically down the concentration gradient. So we can define osmosis as the movement of water molecules from a region of higher water potential to a region of lower water potential through that semi-permeable membrane, which is your cell membrane. Now there are three different types of osmotic solutions or osmosis solutions. There's isotonic, hypertonic, and hypotonic. If the concentration of solute on both sides is the same, it's called isotonic. In this case, we're looking at the concentration of salts. So here I have a beaker representation. Solution is isotonic. I have 10% salt water in the cell. This red little bag looking thing here is the cell. And I also have 10% salt solution outside the cell. So basically I have a net balance or a net zero water movement. So I do have water flowing in and out. There's always going to be water flowing in and out. Nothing's going to stop that. It's a very passive, non-stop action. But the total net gain or loss is zero. So basically as much water moves in the cell, the same amount of water moves out of the cell. And that's isotonic. Iso means same. The next type of solution is the hypertonic solution. So in a hypertonic solution, there's going to be more salt molecules outside the cell, which causes water to be sucked out of the cell. So you can kind of think of it like when you're really hyper and you're stuck inside the house and you're really, really hyper, you want to just run outside and you want to go play. Or maybe it's, your kid's sister or brother that wants to go play. But hyper kids want to go outside and play. So in the case of a hypertonic solution, you're going to have the water molecules leaving the cell, just like this arrow shows, going out into the bloodstream. So if you drink a lot of salt water, for example, let's say somebody dares you and you drink 2 or 3 glasses of salt water, you're not just going to get really, really sick. What can actually happen is as the salt floods into your blood veins, which this beaker here is representing your blood vein, kind of looks like a blood vein. What's going to happen is as the salt rises in your bloodstream, the water in each of your individual cells is going to start leaving the cells, dehydrating the cells, causing them to shrivel up like little raisins. This can cause your cells to die. If you drink too much salt water, eventually this can not only dehydrate you, but it can cause delirium and eventually death. The next type of solution is the hypotonic solution. Hypotonic solution. There's a lot less solute outside the cell than there is inside the cell, so water gets sucked into the cell. The way I remember this one is based on the O here in hypotonic. An O looks like a water balloon. And when you fill a water balloon up too high, it bursts. So hypotonic solution has an O. O looks like a water balloon. In a hypotonic solution, what basically happens is you have a higher ratio of salt in the cell and a lower ratio of salt in the bloodstream. So a real-world example of this would be if you were to go to the hospital to get an IV. Let's say you needed to rehydrate, and so they gave you IV fluids. If the IV fluid bag was pure water, nothing else in it, just pure H2O, that would be a 0% solution of water, so no salt. And what would happen is all of the excess water that was in the solution would go into your bloodstream and it would flood into your salty cells. So eventually what could happen is this could kill you as well. If you had enough pure water put into your bloodstream and it filled enough of your cells, it could cause them to each burst. So basically what could kill you is dying at the cellular level, bursting one blood cell at a time. Pretty painful. So we're going to summarize the three different solutions and what would happen to each of the cells. So first off, this is your isotonic solution. It's the perfect form of the cell. So this is iso. Down here on the left, we've got turgidity happening. So in this case, we have water going into the cell, causing the cell to fill. So what solution would this be? If you have a dilution of water outside the cell, this is hypo. All right, over here on the right, we have a concentrated solution that the cell has been put into. So the water is going to leave the cell. And what happens with the plant cell here is it's a little unique to the plant cell. The plasma membrane will actually pull away from the cell wall. You can kind of see it pulling away here. It's called plasmolysis, or the cell plasmolycete. You don't have to pronounce that. I just thought you might like to know. But that's what happens with a plant cell. The cell membrane actually pulls away from the wall. It kind of shrinks up. As we discussed in animal cells, which is what we use most of our examples with, we have the isotonic cell here. Not even going to try to write that right now. Down on the left-hand side, we have our hypotonic solution. Remember that O forms a water balloon cell. We have water rushing into the cell. The example that we use to this is if you get IV fluids that are just pure water, that would cause the pure water to burst into your cells. Over here on the right, we have the hypertonic solution. Remember, when you're hyper, you want to leave, so the water leaves the cell. And the example that I used of this is when you drink salt water, like ocean water. You have much more salt in your bloodstream, and your cell releases water, or the water passively leaves the cell to try to balance out on both sides. One of the things that I want you to remember before we move on is osmosis doesn't have a thought process. Remember, this is all passive. No one's controlling this. It's simply physics and the way that molecules like to spread out and balance. The next type of diffusion we're going to look at is facilitated diffusion. So if you have charged particles, or if you have really large molecules and they need to move across the membrane, there's another process that has to be found because they're not going to get through the plasma membrane. They're just too big, or they don't have the right charge. These molecules might be also less soluble or even insoluble in the lipid bilayer itself. So they're going to have to be moved through something called a pore. So there's these lined pores in the plasma membrane. These pores are called channel proteins. So channel protein, draw an arrow here. There's your channel protein on the cute little cartoon. So these pores are called channel proteins. These line a water-filled pore in the membrane, so the water-soluble molecules can actually pass through much more easily. There's different channels to allow different substances to pass through. So these channels are selective. They select what they want to let through. Some channels are also gated, which means they will only open if they're properly stimulated or induced to open. Let's look at a specific one. An example of this facilitated diffusion is facilitated osmotic diffusion. If a lot of water needs to get through the membrane all at once, they may need to pass through a channel protein known as an aquaporin. These are basically, they're for massive passive diffusion, but is also considered facilitated. So the water has to use a door basically because there's too many water molecules going through at once. There's still not an energy required, there's no energy needed, so you can see that the water is simply flowing through the larger channel. And again, anytime you use any type of mechanism besides the membrane itself, it's going to be called facilitated diffusion. I kind of think of it like when you have to go to the bathroom, and if you're being polite, you might say, I have to use the facilities. You're facilitated through an opening to go into it, and that's basically what facilitated diffusion is. And before we move on, let's summarize the types of passive transport. First, there's diffusion. Molecules are going to go right through the plasma membrane. And then there's facilitated diffusion. Molecules require a doorway or a channel protein to allow them to pass through. Neither of these require energy, It's a natural movement in physics of the molecules themselves, going from an area of high concentration to an area of low concentration in the direction of the concentration gradient. In passive transport, there are three types of diffusion that only move water. There's the hypotonic solution, where water rushes into the cell to balance out the salt concentration. You have less salt outside the cell than you have inside the cell. Then you have your isotonic solution, where it's balanced, same concentration on the outside as on the inside. And then the third one is your hypertonic solution. In the hypertonic solution, you have much more salt concentration on the outside, and so the water rushes out of the cell. So sometimes substances need to be moved from where they are at a lower concentration to where they're at a higher concentration, or against the concentration gradient. This is going to allow cells to take up essential molecules even when they are at a low concentration outside. Because molecules are moved against the concentration gradient, it's going to require energy. This energy is going to be in the form of ATP. This form of transport is called active transport, and it's going to use carrier proteins similar to those involved in facilitated diffusion. So if there's really large groups of molecules or a large molecule and it needs to enter or exit a cell, they're going to do this using vesicles. The material to be transported out of the cell is going to get surrounded by the plasma membrane. The vesicle is going to fuse with the cell's surface, the surface of the membrane, and then the contents are going to leave. This is called exocytosis. You can kind of think of it like exiting the cell, exocytosis. And then there's endocytosis. If materials are going to enter the cell, they're going to do so through the plasma membrane as well. The plasma membrane is going to surround whatever the material is. The membrane is going to seal off to form a vesicle, which is then going to be moved into the cell. This is called endocytosis. And you can kind of think of it like endo, going in through the door, endocytosis. So if the material is a fluid, these little minute vesicles are going to form. And this type of endocytosis is called penocytosis. That's of liquids. If the material is relatively large, but it's solid, like let's say a digested material, and it's needs to be digested by enzymes, after fusion of the vesicle with the lysosome, it's going to be called phagocytosis. And here it is over here. Phagio means to eat. So this is going to occur in white blood cells that are going to be ingesting bacteria and other foreign bodies. So let's summarize. First, we have passive diffusion. In passive diffusion, it's all about balance. Things will either go directly through the membrane or through a protein channel, but there's not going to be any energy. The second thing we talked about is water diffusion. Water diffusion is through osmosis, and only water will move. It's going to go through the membrane if no channel is present, and through a protein channel if it is there. Third, only a few things can pass through a membrane: water, oxygen, and carbon dioxide. This is necessary for cellular respiration and photosynthesis to take place, and we're going to talk about this in just a moment. 4. Larger particles must use the facility or the protein channel. This is called facilitated diffusion. The protein channel does not need energy if the particles are going in the direction of the concentration gradient. This means they are balancing on both sides. In some cases, if molecules go through the channel against the concentration gradient, it will be considered active transport. It's still facilitated, it's just active too. And it will require ATP. This means the sides will not be balanced and energy is required in the form of the ATP molecule. So now we're going to look a little bit closer at the energy. So we've been talking a lot about the exchange of gases through cells and the cell membranes. But do you remember what the word for passive exchange is? All right, so cellular respiration is going to occur in the mitochondria, as pictured here. And the mitochondria is actually a membrane-bound organelle, so it actually has a plasma membrane of its own. This process is in your bodies, and it's a way of creating chemical energy. This energy is in the form of ATP, which is also called the energy molecule. So you can kind of think of this process in the same way that you would go through your daily routine of eating and living and breathing. So for example, let's say you were going to go for a run. So before you go on the run, you might want to load up on carbohydrates, which are really high in the sugar glucose. This is actually produced by plants, by the way, through photosynthesis. As you go through your run, you would start to inhale very deeply as your body needs more oxygen. The more oxygen you take into your cells, the more glucose your cells absorbed. So here's the glucose going in and the oxygen going in as you go for your run. You're inhaling the oxygen, and you also already ate the glucose in the form of, let's say, a pasta. So the more oxygen you breathe in and the more glucose you load it up on, the more your cells are going to be able to cellular respirate. As you go through your run, you would start to inhale heavily as your body needs more oxygen. And like I said, the more oxygen you take in to your cells, the more glucose your cells absorb, the more energy your mitochondria will be able to create on your run. So your body will exhale the carbon dioxide, it's shown here, and your cells will actually release a tiny bit of water. We can go through the exact molecule ratio in just a moment. But they're actually going to release water, and you're going to exhale carbon dioxide. But if you look here at the chloroplast, these are the ingredients that the chloroplast needs in order to carry out photosynthesis. So on the flip side, a plant would suck water up through the roots. It would then inhale carbon dioxide, and through light energy hitting the chloroplast and reacting, and basically as the light energy goes into the chloroplast, it kind of releases these electrons that kind of bounce around like a ping pong ball, and it releases these electrons, creating a chemical reaction itself. And this is going to create the glucose energy, and that energy is going to be what you eat to prepare for your run. And the plant also exhales oxygen in the process through the stomata, which are the tiny little pores on its leaf. And so basically the entire process, photosynthesis to cellular respiration, is one big circle. Keep in mind that plant cells also have mitochondria, so they can carry out both of these processes, cellular respiration and photosynthesis, in their cells. So go ahead and write down the formula for both photosynthesis and cellular respiration. Feel free to pause the video while you do this. The purpose of photosynthesis is to use carbon dioxide and water with sunlight or radiant energy to make sugar or food or glucose. The byproduct of this reaction is oxygen. The purpose of cellular respiration is to make cellular energy, or ATP, by breaking down that glucose with the oxygen. The byproducts are carbon dioxide and water. One process couldn't exist without the other. It's a full circle of a formula. Cellular respiration and photosynthesis, as I mentioned, are part of the carbon cycle. Here pictured A and B are the outputs of photosynthesis and the inputs of cellular respiration. So I challenge you now to identify what are A and B. Feel free to pause the video and go back to see if you missed anything.